Usually when we talk about getting a medical diagnosis, there's this this underlying expectation of precision.
Right, like finding the exact broken part.
Exactly. Like if you shatter your femur, the X-ray shows that jagged white line, and the orthopedic surgeon just points right at it and says, there's the problem.
Yeah, it's incredibly localized. It's binary. It's either broken or it isn't, right? The mechanics are visually indisputable.
Completely. But what if I told you that in the world of cancer genetics, finding that exact localized break like the specific mutation is actually, it's only half the story.
Oh, easily. Maybe even less than half.
Right. What if the exact same genetic mutation that causes this devastating, rapidly spreading fire in one part of your body is, a little more than a harmless flicker in another part?
Which is wild to think about.
It is. What is the biological neighborhood where that mutation lives changes absolutely everything about how it behaves.
And that right there, that represents a massive paradigm shift. Because for decades, we have been so conditioned to view cancer as a purely gene-centric problem.
Yeah. Find the bad gene, fix the problem.
Exactly. The prevailing logic has always been you sequence the tumor, you find the bad gene, you deploy a targeted therapy, and you stop the cancer.
Right.
But the research we are unpackaging today, it just completely shatters that simplicity.
And it shatters it with just an overwhelming amount of data.
Yeah.
So today's deep dive is centered on a monumental preview article from the journal Cancer Cell.
Yeah, authored by Maria Kalaba and Nicholas McGranahan.
Right, and they are analyzing this, frankly, unprecedented study by Bandlamudi and colleagues.
Yep.
So our mission today is to dig into how analyzing over 50,000 tumors proves beyond a shadow of a doubt that cancer requires a context-aware framework.
We can't just look at the gene anymore.
No, we have to look at the zip code. Okay, let's unpack this. Because to truly understand the neighborhood, we first need to look at the scale of the census they conducted on the residents, right?
Yeah, let's talk about the data set, because this wasn't just, a few hundred samples in a local lab.
Not at all. They evaluated cancer-type specific patterns across 54,331 tumors.
It's a staggering number.
It really is. And that massive cohort spans 64 broad cancer types. And when you drill down into the clinical specifics, that's 448 distinct cancer subtypes.
The statistical power there is just undeniable. But, what makes this data incredibly robust isn't just the volume.
Right. It's how they processed it.
Exactly. It's the uniform processing. So they used targeted sequencing panels looking at up to 505 specific genes. But crucially, they paired this with matched normal samples for all the patients.
Wait, explain the matched normal part real quick, just so we're all on the same page.
Sure. So by comparing the tumor DNA directly against the patient's own healthy baseline DNA, they eliminated massive amounts of background noise.
Oh, I see. So it filters out the genetic quirks you were just born with. Precisely.
It allowed for highly accurate identification of what was truly an acquired disease-driving variant, rather than just a benign, inherited quirk.
And because they had this massive, pristine data set, they were able to uncover 164 novel mutation hotspots.
Right. These specific, highly recurrent genetic sites.
Now, individually, I know these hotspots are relatively rare, but the paper notes that when you aggregate them, 5% of all the tumors in this massive cohort harbored one of these novel hotspot mutations.
Which represents a massive therapeutic target pool.
It does. But what really jumped out at me in the paper is when these mutations occur, because they describe these new hotspots as early and truncal.
Yes, truncal. The concept of truncal mutations is foundational here. It refers to the MRCA, or the most recent common ancestor.
Okay, so the way I visualize this, and let me know if this analogy holds up, is like looking at a massive sprawling family tree.
Okay, I like where this is going.
But it's a family tree of rogue cancer cells inside a single tumor.
That's a perfect way to visualize it, yeah. At the very bottom of that tree, you have the thick base trunk.
Right.
And that represents the most recent common ancestor. It's that original cell that crossed the line into malignancy.
So it's the starting point.
Exactly. And as the tumor proliferates, it branches out. Different cells acquire distinct new mutations due to environmental pressures, creating this complex canopy of divergent branches.
But no matter how wild or chaotic those branches get at the top of the canopy, every single one of them still connects back to that original trunk.
Right, meaning a truncal mutation is present in every single cancer cell in the entire tumor. It's an early disease-initiating event.
And the paper goes a step further, right? Pointing out a specific mechanism for how these truncal hotspots operate. It notes they predominantly occur in tumor suppressor genes.
Yes, and they show a heavy enrichment for loss of heterozygosity, or LOH.
Yeah, I was struck by how these truncal hotspots heavily favor this loss of heterozygosity. It's like the cancer isn't just relying on a broken gene, right?
No, it's actively seeking out and eliminating the backup copy.
Exactly. It reminds me of the dual redundancy systems in cars or airplanes. Like you have two brake lines. If a rock snaps one, your first hit, you don't crash.
Right, you still have the backup brake line keeping you safe.
Right. So the cancer needs that second hit to cause a crash.
And that is exactly the two-hit hypothesis in action. Tumor suppressor genes are your cellular brakes, preventing uncontrolled division. You inherit two alleles or copies of each gene.
So losing one function isn't enough to cause a malignancy?
Precisely. The loss of heterozygosity is that critical secondary failure. The cancer actively drops the remaining functional backup, leaving the cell completely blind to its own rampant division.
So if we pause here for a quick subtopic recap, just to synthesize what we've covered in this first section.
Always a good idea.
Okay, bullet point 1. We are looking at a study of over 54,000 tumors that found 164 novel hotspots.
Right.
Bullet point 2. These new hotspots are truncal, meaning they are early, disease-initiating events at the base of the cancer family tree.
Exactly.
And bullet point 3, they largely occur in tumor suppressors and require that two-hit loss of heterozygosity to fully disable the cell's brakes.
Perfect summary.
So a question for you listening right now. Think about that cancer family tree. If you were a scientist with limited funding, would you focus your research on trying to poison the trunk? Or would you try to build a net to catch all those rapidly mutating branches?
That is the central tension of modern oncology. In theory, if a target is truly truncal, a drug that effectively attacks it should eradicate every single cancer cell in the patient's body.
Which is the holy grail.
It is. But as Kaleva and McGranahan point out, the behavior of these genetic drivers isn't always so rigidly predictable.
Right, which leads us to the most fascinating pivot in the paper, the canonical versus non-canonical context, the whole where and when of it all.
This is where the researchers really categorize the genome based on its biological zip code.
So define canonical for us first.
Sure, a canonical gene is one that is significantly enriched for disease-driving mutations within a specific tissue type. It's the usual suspect. You expect to see it there.
Okay, and so a non-canonical gene is an alteration found in a tissue where it's not typically associated.
Exactly. And the data from Banlamudi is stunning here. 32% of driver events, nearly a third of all the mutations actively pushing the cancer forward, happen in these non-canonical genes.
Wait, nearly a third? That is a huge proportion of cancer drivers just operating outside their normal zip codes.
It really is. And it fundamentally alters the evolutionary timeline. When a driver mutation occurs in its canonical context, its usual tissue, it typically acts exactly as we discussed.
As an early truncal event.
Right. It's a clonal event present in all the resulting cells.
To use a corporate takeover analogy, a canonical driver is like the rogue CEO. They are plotting the hostile buyout from day one. They are the trunk of the conspiracy.
I love that. But a non-canonical driver isn't the CEO.
No, they're like a mid-level manager who sees the takeover happening halfway through the year, opportunistically jumps on board, and steals data to accelerate the process right before the deal closes.
That is a highly accurate way to visualize it. Non-canonical drivers tend to emerge as late subclonal events.
So they join the moving bus later on.
Exactly. They are late to the party, but they provide a sudden, massive evolutionary advantage to one specific branch of the tumor. The paper highlights the PIK3CA gene to illustrate this beautifully.
Oh yeah, walk us through the PIK3CA mechanics, because this completely blew my mind when I read it.
Okay, so in breast, uterus, and bladder cancers, a PIK3CA mutation is highly canonical. It is expected. And true to its canonical nature, in those specific tissues, it is predominantly a clonal early truncal event. It's your rogue CEO initiating the disease.
Right.
But if you look at lung adenocarcinoma or glioblastoma, a type of aggressive brain cancer, PIK3CA mutations are strictly non-canonical.
And so they behave differently.
Completely. The data clearly shows that in the lung and the brain, this exact same genetic mutation appears almost exclusively as a late subclonal event.
But structurally, why would that happen? I mean, the DNA sequence of the PIK3CA mutation is identical, whether it's in the breast or the lung, right?
It is identical, yes.
So why is it the mastermind in one organ, but merely an opportunistic mid-level manager in another.
Because the selection landscape of a tumor shifts wildly depending on the tissue. The epigenetic rules like how tightly DNA is wound, which genes are accessible, are completely different in breast tissue compared to neural tissue in the brain.
Oh, that makes sense. The local environment dictates what tools are useful.
Exactly. Plus, the local immune pressures vary. In a non-canonical setting, a PIK3CA mutation might actually be detrimental or simply useless to a cell early on.
So it just sleeps in the back seat.
Right. But as the tumor grows, creates its own blood supply, and changes the local microenvironment, those early constraints might relax.
So suddenly, a gene that was useless in the early days of a lung tumor becomes the exact tool a specific subclone needs to survive, say, a lack of oxygen later on.
Precisely. It's entirely dictated by the shifting neighborhood.
Wow. Okay, let's do another quick recap for everyone listening.
Sounds good.
Bullet point 1, canonical drivers are early expected trunk events.
Right.
Bullet point 2, non-canonical drivers are late unexpected subclonal events, and they make up 32% of all drivers.
Which is a huge chunk.
Bullet point 3, context changes everything. PIK3CA is an early driver in breast cancer, but a late addition in lung cancer.
Spot on.
So my question for you, the listener, is this. If a mutation's behavior completely changes based on its biological zip code, how should we rethink the way we name and classify cancers?
It's a massive debate. Should we still just call it lung cancer or classify it by its evolutionary timeline?
Exactly. And speaking of timelines, let's jump into section 3, because this tissue context doesn't just impact the tumor's growth, it deeply impacts the clinical presentation of the patient, specifically regarding age and immune response.
Yes, because this study wasn't just sequencing cells in a vacuum. They correlated the genetics with incredibly rich clinical data, including the age of the patients at diagnosis.
And what they found regarding cutaneous melanoma skin cancer was stark. They compared tumors driven by BRAF fusions to those driven by standard BRAF point mutations.
Right, so a gene fusion is a massive structural rearrangement. Two completely separate genes break apart and fuse together inappropriately, creating a hyperactive hybrid gene.
And the study found that melanomas driven by these BRAF fusions are associated with significantly younger patients compared to those with standard point mutations, or SNVs.
Yes, and mechanically, it points to fundamentally different mechanisms of tumorigenesis based on the strength of the driver.
Wait, why would a fusion bypass the immune system of a 30-year-old, whereas a point mutation usually takes decades of sun exposure to finally trigger the disease in a 60-year-old?
Well, a fusion is often a much more potent, constitutively active oncogenic driver. It's like permanently wiring the accelerator to the floorboards.
So it just overwhelms the system.
Exactly. It requires far less time and fewer subsequent mutations to overcome the robust immune surveillance of a younger patient. A point mutation might just be a sticky accelerator pedal that requires decades of additional cellular mistakes to finally cause a crash.
And speaking of that robust immune surveillance, that transitions us perfectly into the most crucial warning in this entire paper, the puzzle of precision immunotherapy.
Specifically T-cell receptor or TCR therapies.
Right. To truly grasp the clinical frustration outlined here, we have to understand the exact mechanism of how the immune system sees cancer in the first place.
It's fascinating biology.
I like to use a shop window analogy for this. Imagine every single cell in your body is a small shop. On the exterior surface of the cell, there are specific molecular display windows.
Right, and in biology, these windows are called HLA molecules, human leukocyte antigens.
And inside the shop, the cell is constantly breaking down proteins. As it recycles them, it takes little peptide fragments and places them into the HLA display window for the immune system to inspect.
Right, and the immune system's T cells are the beat cops walking the neighborhood.
Exactly. They stroll past the cell, look into the HLA window, and if they see normal, healthy protein fragments, they tip their hat and keep walking.
But if the cell is cancerous, it's producing mutated proteins, which we call neopeptides.
And if the T-cell cops see those bad goods in the window, they recognize the cell has gone rogue and they destroy the shop.
That mechanism is the entire basis for TCR therapies. We can genetically engineer a patient's T cells to specifically recognize the exact neopeptides created by famous cancer hotspots.
Yeah, figure 1 of the paper highlights this, noting highly immunogenic mutations in KRAS, like the G12D and G12V variants, and TP53 mutations like R175H, R248W, and Y220C.
And the data says 13% of patients have these specific, highly targetable hotspot mutations.
which on paper sounds incredible. We know exactly what the bad goods look like, so we just send in the engineered cops.
But the paper reveals a brutal mathematical catch, a truly devastating catch.
What is it?
Of that 14% who have the mutation, only 22% of them possess the specific HLA allele required to actually present those peptides to the immune system.
Wait, because of HLA restriction?
Yes. The display windows aren't a universal shape. Let's say the mutated KRAS neopeptide is shaped like a triangle.
Okay.
If you, based on your genetic inheritance, only have HLA windows designed to hold square proteins, you physically cannot display the mutated triangle.
Oh, wow. So the bad goods are inside the shop, but they never make it to the window.
Exactly. The engineered T-cell cops walk right on by, completely blind to the cancer. Even if a patient has the perfect targetable mutation, if they don't have the restricted HLA allele, the multi-million-dollar TCR therapy is useless.
And to complicate this on a global scale, the paper notes significant ancestry-specific differences in these HLA alleles across different populations.
Meaning certain immunotherapies might be inherently biased towards specific demographics. It is incredibly frustrating for clinicians.
But what's even more alarming is how the tumor actively fights back against the windows that do work.
Yes, immune escape.
The study found that 25.4% of tumors exhibit a copy number loss of HLA.
The tumor senses the evolutionary pressure from the T cells, realizes the cops are looking at the window, and simply deletes the genetic code for the window entirely.
They literally brick up the storefront.
Precisely. If there is no HLA expression, there is no antigen presentation. The cell becomes functionally invisible to the adaptive immune system.
That is terrifying.
And Kaliva and McGranahan point out that tumors also employ non-genetic mechanisms of HLA downregulation, epigenetic silencing, basically, to turn off the window without even deleting the gene itself.
Okay, let's take a breath and recap this intense immune section.
Good idea.
Bullet 1. Gene fusions cause earlier onset disease than point mutations because they're stronger drivers.
Right.
Bullet 2. Only 22% of patients with targetable KRAS or TP53 mutations have the right HLA window to actually show them to the immune system.
Deadly, yes.
Bullet 3. Over a quarter of tumors actively delete or silence their HLA genes to hide from the immune system entirely.
Nailed it.
So, listener, if a tumor is actively destroying its own HLA display windows, how might scientists force those windows back open? Because without a display, the therapy is completely blind.
Which brings us to the limitations of our current approach and the roadmap this paper provides for the future.
Section 4. Let's get into it. Because as comprehensive as a 54,000 tumor dataset is, the technology they used has inherent blind spots.
Right. They relied on targeted panel sequencing, looking at up to 505 genes, which is incredibly deep, but narrow.
It's like investigating a massive dark mansion by only looking through the keyholes of 505 specific doors.
That's a perfect analogy.
You get a crystal clear view of the main furniture in those specific rooms, but you are entirely blind to what might be lurking in the hallways, the attic, or, the unmapped corners of the house.
Exactly. A targeted panel is phenomenal for finding known hotspots, but you are ultimately only looking at the genes you preselected. The alternative is whole genome sequencing, or WGS.
Which would be like turning on the master light switch for the entire mansion.
Right. Because the Bonla-Moody study utilized targeted panels, it inherently misses certain structural variants and strictly limits the discovery of de novo brand new, uncharacterized cancer driver genes.
But the authors don't just point out the dark rooms, right? They map out how we turn on the lights moving forward. The paper emphasizes that the future is multi-omic data integration.
Yes. We can no longer rely solely on genomics, the raw DNA code. Figure 1 of the preview article clearly advocates for an integrated approach combining genomics, epigenomics, and the immune context.
Okay, so sticking with the house analogy, if genomics is the structural blueprint of the biological mansion, telling you where the walls are,
then epigenomics represents the electrical system, the complex array of light switches and dimmers that dictate which rooms are actually functional at any given moment.
Oh, I like that. And the immune context.
That is the neighborhood watch outside, constantly interacting with the house and forcing the occupants to adapt. To truly understand the clinical impact of a single mutation, you must analyze all 3 omics simultaneously.
Though, to be fair, the authors do highlight one fascinating exception where tissue context seems to be overridden.
Yes, to provide a balanced view, we should mention tumors driven by mismatch repair deficiency.
Wait, what kind of tumor manages to ignore its own neighborhood?
Well, these are hypermutated tumors where the DNA repair mechanisms are so utterly broken that the cell accumulates thousands of mutations. In these specific cases, immune checkpoint blockade therapies are highly effective regardless of the tissue type.
Why is that?
The sheer volume of neoantigens is so loud, the immune system hears the alarm whether the tumor is in the colon or the lung.
So the noise just overwhelms the local zoning laws.
Precisely. But that is a specific exception. For the vast majority of malignancies, the analysis of these 54,000 tumors unequivocally proves that tissue context is the supreme dictator of tumor behavior.
So final quick recap. Bullet 1. Targeted panels of 505 genes are great, but they miss the full picture provided by whole genome sequencing.
Right.
Bullet 2. The future is multi-omics, combining DNA, epigenetics, and the immune environment.
Exactly.
So listener, think about the omics. If genomics is the blueprint and epigenomics is the light switches, what role does the immune system play in your biological house?
It demands a more holistic biology. We have to abandon this purely gene-centric view.
Bringing all these threads together, it really shows that our entire philosophy of precision medicine has to adapt. We spent the last 2 decades obsessing over the gene. Find the broken part, fix the broken part.
But the reality exposed here is that a mutation isn't inherently good or bad in a vacuum. The exact consequence of a genetic alteration depends heavily, and sometimes entirely, on the biological neighborhood in which it arises.
It's incredibly relatable when you think about it on a human level. Just as a person's behavior, their trajectory, their choices are heavily influenced by the community they live in and the local stresses they face, a gene's behavior is dictated by its tissue.
It's the interactions in the shifting environment that drive disease progression, not just the isolated molecular components.
Which leaves me with a thought I want to bounce off you, the listener, as we wrap up this deep dive. If we know that the exact tissue context dictates the evolutionary timeline of a tumor, if the zip code dictates whether a mutation acts as an early mastermind or a late-arriving opportunist, could we one day use advanced AI to map out a cancer's entire future evolutionary path?
That is the big question.
Think about how predictive algorithms map out traffic patterns based on city infrastructure. Could a system look at the omics of a single biopsy, analyze the biological zip code, and perfectly predict every single evasive maneuver that cancer is going to make before it even happens?
If we can master the context-aware framework, predicting the evolutionary future of a tumor is the ultimate horizon of precision oncology.
Something to ponder as you look at the complex neighborhoods around you.